Inside of the heart, there are very specialized cells known as pacemaker cells. And these pacemaker cells are responsible for the spontaneous sort of depolarization of the cardiomyocytes and the contraction of the heart. So it's for these cells specifically that the heart can continue to beat even if we take it outside of the body, which is pretty cool. Now, these specialized cells set the rhythm for the heart, and they are autorhythmic, meaning that, they don't receive any sort of neural input. They just spontaneously depolarize. So sometimes people will refer to these as myocardial autorhythmic cells. Now, the general people tend to just refer to them as pacemaker cells because it's descriptive, it's what they do, right? But anatomists and physiologists, of course, will use the big long name, which is myocardial autorhythmic cells. All right, now these autorhythmic cells are very special because they are modified cardiomyocytes. Okay, so that's where the myocardial comes from. So I'm just going to write that down so that we know that. These are modified cardiomyocytes. And remember, a cardiomyocyte is a cell, muscle cell of the heart. So these are modified muscle cells. These are not nervous tissue. This is cardiac muscle tissue. All right. Now, the way these cells work is actually pretty dang cool. So if we look and we map over time, we're going to map our membrane potential. And I'm just going to put MP. And again, I'm not going to draw numbers because I don't want anybody to be concerned with the numbers, right? The concept here is what's important. So let's say this cell starts down here at rest. We have our resting membrane potential. Okay? What's going to happen is, let's say we generate an action potential. Let's say threshold is right here. So if you don't know what threshold is, go back and rewatch some videos in the nervous tissue. Right? But let's say we reach threshold and we generate an action potential inside of this cell. Okay? Now remember that the cell is going to repolarize and then it's going to hyperpolarize, okay, before turning to rest. That state there where we go below resting membrane potential, that's your hyperpolarization. Now these cells have very specialized channels in them. They're called funny channels. Okay? They're called funny channels because the scientist who described them said that the flow of current was really funny. It didn't understand how it worked. We now understand how it works, but back then they didn't know initially, so we called them funny channels, and the name's kind of stuck. But at this hyperpolarization, those channels open up, and they are cation channels. They're permeable to potassium, but they're really permeable to sodium. And so what happens, because they are permeable to sodium, the equilibrium potential of the cell, once they open up, let's say they open up at time blue, right? They open up, the membrane potential will creep back towards threshold, like so. Now at threshold, what's going to happen? You're going to generate an action potential, okay? Because here, inside of these specialized cells, there's another channel that's going to open up, and these are going to be voltage-gated calcium channels. So this is different than the standard membrane potential and action potential that we talked about, right? So these are voltage-gated calcium channels, right? And that cell's going to depolarize, and then it will repolarize, and then it will hit that state of hyperpolarization. And at hyperpolarization, those funny channels are going to open back up and the cell will creep back towards threshold, and then it will repeat the process. And as long as it generates an action potential and the cell hyperpolarizes, these funny channels will open back up, the cell will move back towards threshold, and the process repeats. Okay, so this is the spontaneous depolarization. This process here is the spontaneous depolarization, which is caused by those funny channels. Now, we now know how those channels work, and they're actually voltage-gated channels that are activated at this voltage, this hyperpolarization voltage. So we refer to these as hyperpolarization activated, and they are of the cyclic nucleotide family. they transport cations. So we call them hyperpolarization activated cyclic nucleotide cation channels, or some people will abbreviate this HCN channels. Okay, so the cell is going to hyperpolarize, the HCN channels open up, and the membrane potential moves towards threshold. At threshold, voltage-gated calcium channels open up, the cell depolarizes, and action potential is generated. And then voltage-gated potassium channels open up, just like in a regular neuron. The cell repolarizes, then it hyperpolarizes, and at hyperpolarization, those hyperpolarization activated cyclic nucleotide cation channels open up, and the process repeats itself. So once we generate one action potential, boom, action potential is generated until the cells die. All right, now these pacemaker cells, or these myocardial autorhythmic cells, are located in very specialized regions inside of the heart. So remember, our heart has two chambers that receive blood. You have a right atrium, and you have a left atrium, like so. Okay, then you have two chambers that send blood. You have a right ventricle, and you have a left ventricle. And we'll say this is mammal, so it's all separated completely. Okay, so these specialized pacemaker cells are located in specific regions. Okay, you have one set of cells up here that is called the SA node, the sinoatrial node. Okay, and so it is sort of the first to fire. So if we look at the firing of the action potentials, right, you have the SA node, sort of will go like this. Like that, okay? Then you have another set of those cells, pockets of those cells. This is called the AV node, okay? It's AV node because it sets at the junction between the atria and the ventricles, the AV node. Now, it will have this spontaneous depolarization as well, like so, but it's a little bit slower. Notice that it's a little bit slower than the SA node. Okay, so because of that, the SA node is the one that sets the pace, and the signal is sent down to this AV node. Now, when that signal is sent down to that AV node will kind of go like this, and then boom, it gets that signal and reaches threshold like this. Okay, so this is if signal from SA node. Okay, in green, what we have is no signal from the SA node. Okay, so this AV node fires a little bit slowly, right? And what happens is it's going to get the signal from the SA node. So the SA node is going to fire, and the signal is going to come down from the AV node. But in the event that this SA node stops firing, look here, that AV node will eventually fire. The heart rate will drastically drop if that takes place, but we do have a backup mechanism if this thing sort of fails. Now, this signal will come down through a junction known as the bundle of His, and then it sort of splits into left and right portions, which we call the bundle branches. Okay, and the bundle branches continue to the apex of the heart, the base of the ventricles, and then they flip around and they come up the lateral aspects of the ventricles, like so. And there's like little sort of pieces that come off of there, and so these here, these are known as the Purkinje fibers. Okay, so the signal will go from the SA node, travel down to the AV node. Okay, from the AV node, it comes down through the bundle of His, splits into the left and right bundle branches. Then at the apex, flips around and comes up the Purkinje fibers. Okay, and that is the pathway that the electrical signal takes from the heart. Now, as the action potentials are generated inside of these cells here, right, these myocardial autorhythmic cells, they'll spread out to the other cardiomyocytes because they're all connected via those junctions, right? Those intercalated discs. So that's the importance, right? Why these pacemaker cells are not nervous tissue. They are modified cardiomyocytes. So when the cells sort of depolarize and they generate action potentials, that action potential will spread throughout the adjacent cardiomyocytes. So your electrical signal, right, goes from the SA node to the AV node to the bundle of His, through the bundle branches, up through the Purkinje fibers. So if we follow that along, right, and know that the signal is spreading out, the atria will contract, right? Then the ventricles contract downwards to the apex, right? And then the signal comes up. And so this sort of the top lateral portions of the ventricles are like the last portions of the ventricles to contract. Okay? So those are the myocardial autorhythmic cells or pacemaker cells.